Invasive species have become a global ecological and conservation crisis. These are species that are living somewhere outside of their native range, and they have cost the U.S. billions of dollars, and in some areas are becoming even more susceptible to invasive species. That's why it's important that we understand the effects these species have on their new invaded range, and also be able to learn how we can mitigate the effects of these species. For my thesis, I'll be working with one invasive species in the Florida Everglades, and though it may look kind of snake-like, it's actually not the Burmese python that everybody thinks of when thinking about invasive species in the Everglades. But though its size might be small, you can see that its population has increased drastically since its introduction in the early 2000s. And that means that though it might be small, it still could have a huge impact on its local food webs. So for my project, I'll be looking at how big of an impact this invasion is on the ecosystem, and I'll be looking at what kind of environmental factors influence our population trends, whether that's the temperature or water levels, sea level rise, these kind of things, so that way we can anticipate future trends in their population as the climate continues to change. The other thing we'll be looking at is how these species affect some of the native species, such as the sunfish. The sunfish might seem like another small, unimportant species, but it has a big economic impact in Florida. It actually is a large food source for some of the fisheries species, such as snook and tarpon. And so if the peacock eels come in and are eating all of the food from the sunfish, they're no longer going to be able to be food for the larger species like the snook and the tarpon. So in order to kind of figure out how these sunfish might be affecting the sunfish, we'll do a few different things. So first we have to catch the fish, and we do that using boat-based electrofishing. So we take a boat out into the Everglades, and we shock the water, and the fish float to the surface. It sounds kind of painful, but it's only temporary, and it's actually less invasive than regular methods and much more effective. So we are able to catch the fish, and then once we catch them, we actually just take a small tissue sample from them instead of keeping the whole fish. The rest of the fish can go back. And we can use that tissue sample to actually do an analysis of stable isotopes, which sounds like a lot, but basically they're just chemical biomarkers that you can trace back what the fish was eating based on those chemical signatures. So we've got the mangroves and the grasses have a specific signature, and then you can go back and trace that to the fish. And we can see if they're competing. But you know what they say, you are what you eat.